Hello there, welcome to another episode of Rare Diseases. Rare Disease Review is a not-for-profit medical journal organized by students from the University of Toronto, McMaster and Western University. This podcast is created by the branch of Western University. The podcast aims to advocate for rare disease awareness and promote students' involvement in the rare disease community. We appreciate your listening and support today. For further information on the scientific translation of rare disease information, please visit www.rarediseasereview.ca. The content for this podcast is not intended to be a substitute for professional medical advice, diagnosis, or treatment, so please always seek the advice of a physician or other qualified health provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay seeking it because of something you have heard from this podcast. My name is Song and you're listening to the Rare Disease Podcast. For this episode, we're going to learn about the incredible progress in genome research in rare disease initiated by the Care4Rare Research Consortium and PhenomeCentral. The research program is called Matchmaker Exchange, which is a collaborative initiative that facilitates international data sharing for matchmaking purpose. This research has an outcome of over 1,500 matches made through the Matchmaker Exchange for rare disease gene discovery in the past 2 years at Care4Rare Canada and 7 years at the PhenomeCentral. Today we have the honor to invite Genetic Counsellor and researcher Matthew Osmond to speak to us about the amazing research that has been done in Care4Rare and PhenomeCentral. Matthew Osmond works with patients, clinicians, and scientists to publish many journal articles regarding rare disease research, which helps to find cases of the same unnamed disorder and allowed collaborations to better understand the underlying mechanism of rare disorder. His contribution to the rare disease community is highly valued, and I can't wait to learn about the matchmaking exchange from him today. Without further ado, let's welcome Matthew Osmond to our podcast. Thank you so much for joining us, Matthew, and it's such an honor to have you with us today.
Nice to meet you, Song, and thank you very much for having me.
So to start, could you tell us a little bit more about yourself and Care4Rare organization?
Sure. So you covered some of the basics already. So again, my name is Matt Osmond, and I am a genetic counselor by training. I've worked as the research coordinator for the last 4 years for the Care4Rare Canada research program, based out of the Children's Hospital of Eastern Ontario Research Institute in Ottawa, Canada. In terms of the Care4Rare organization itself, so Care4Rare is a Canadian research program which aims to identify new rare disease genes for undiagnosed patients. So although I'm personally based out of the CHIO Research Institute here in Ottawa, the program actually consists of over 300 clinicians, bioinformaticians, scientists, and researchers from 21 different academic sites across the country. So it's really a big collaborative effort to study rare diseases because, as we know, there are so many where we still don't understand the underlying genetic cause yet.
As I have learned, you carry the title Rare Disease Matchmaker, which is a very interesting name. Could you explain what Matchmaking Exchange means and why is it an important process for rare disease research?
Sure. So what I can do is I'll give a bit of an introduction to matchmaking as a concept in general, and then I can sort of explain why it's so important for rare disease research. So matchmaking is really one of the many tools that researchers have to try and approach research, especially with regards to rare diseases. So when we perform genetic analyses for undiagnosed rare disease patients, sometimes we find a change, or what we call a variant, in a gene that hasn't been associated with a human disease before. And so for us in research, the major question is, how do we try and go about proving that this new gene is associated with an undiagnosed patient's rare condition? So one of the most common ways we try and gather evidence to sort of prove this association is we try and find other rare disease patients out there in the world who have a similar set of clinical features. So you'll hear me call those clinical features phenotypes, and a similar type of variant in the same gene. So finding these similar patients out there in the world, in general, is called matchmaking. So previously, matchmaking was a very manual process. It was all sort of based about who you knew in the scientific world. People would go to case conferences, they'd review recent literature, and really it was just about contacting your colleagues by email or phone, meeting up with them in person and saying, hey, I have a patient with a variant in a new gene. Do you know of anyone else who has a similar patient to me? As digital systems became more common, several databases were created to store the data of rare disease patients. The major problem there was a lot of people had sort of this great idea at the same time. So a lot of these rare disease databases kind of sprung up in a similar time period, but originally these databases weren't actually able to exchange information with one another. And for matchmaking, so identifying these similar patients, you really need to be able to look at as many sources of information as possible. So to fix this issue, the Matchmaker Exchange was launched back in 2015. And so the Matchmaker Exchange is basically a big collaborative effort, a network that connected multiple rare disease databases to one another to allow them to basically communicate. Today, the Matchmaker Exchange network consists of 9 different rare disease databases and 2 knowledge sources from across the world, and collectively share over 120,000 cases across the world.
That's fascinating. When you talk about the previous way of finding, I guess, matchmaking rare disease patients, it just reminds me of finding Wordle, the children's book.
Yeah.
It must be really hard. And it sounds like this is very useful and saves a lot of time and effort between teams.
For sure. Yeah.
It's amazing. Coming from a patient's perspective, what would be the targeted group in need for this kind of matchmaking?
So I think currently, when I think about who is, what kind of patients are involved in matchmaking, the main group I think of are patients who have a rare disease that is suspected to be caused by a singular genetic change, or variants in a single gene, so what we call a Mendelian condition. And they're individuals who haven't had a genetic diagnosis after their clinical testing. So clinically, they're undiagnosed and they've consented to be part of research for rare diseases. For these individuals who are undiagnosed, as I mentioned, generally they'll have a variant in a gene that hasn't been associated with a human disease before. Sometimes they can also have variants in a gene that is known to cause a human condition, but it may be that we're looking at a patient who has a different set of features from what's known in the literature, or maybe a different set of variants from what's known in the literature. So for example, a known condition may be associated with one variant in this particular gene, but we have a patient with a different presentation who has 2 variants in that gene. So those are some of the examples for when matchmaking can be useful to find similar patients.
It's really interesting. So if a patient that wants to be in the program, how can they submit this genetic information to the Matchmaking Exchange?
So I would say for the most part, being involved in programs like Care4Rare that submit information to the Matchmaker Exchange, it involves being consented to rare disease research specifically. So often what we tell people who come up to us and they say, I'm interested in being involved in rare disease research. Often it's, talking to their clinician and seeing, does it make sense to have a referral to see a geneticist to see if this kind of rare disease research would be useful. In terms of how data gets submitted to the Matchmaker Exchange, as I mentioned before, there's many different rare disease databases that are connected through the MME. And so it's simply a matter of once researchers are sort of involved with a given database, they can contribute new information to that database. The exact data depends on the database that you're contributing from. So some databases require just the name of a gene to participate in matchmaking. and some are more strict in terms of the information that you need. So you might require more information on a patient's clinical features or more information on their specific variant that you're interested in.
So it sounds like the first point of reaching out is through the clinicians of these patients then?
Yeah, to explain that, I would say for patients who are interested in getting involved in rare disease research, that really needs to be done by speaking to their clinician, seeing if a referral to genetics would be appropriate, and then from there seeing if the geneticist is interested in helping participate in rare disease research. But the second part of my answer is more talking about, once a patient is in a research program, how does their data get contributed to matchmaking?
Wonderful. So in the research program, I saw that a huge advance in data sharing. So I'm just curious, how important is data sharing in terms of benefiting the rare disease community?
Yeah, I mean, data sharing in general is really, really important, not just for matchmaking, but for rare disease research as a whole. And the major reason for that is because when these conditions are so rare, it's really difficult to sort of find patterns within rare diseases, or to gather enough information on a particular condition. It's really difficult to get that information from a single institution or source. When we see people with undiagnosed rare conditions, they may be the only person that we know of in the city, in the province, or even the country that has that exact presentation of features or a variant in a specific gene that we're interested in. So for that reason, we really need to be able to data share across borders. Being able to identify patients who are from other countries, other continents even, is a big part of being able to find those connections to drive research forward.
That's amazing. It really takes more than a village. It takes the entire globe to work together to help the community.
Exactly.
So how exactly is I guess I'm asking the next step, which is after the data is being shared, how is the match made?
Sure. So to keep it basic, after a case is added to a rare disease database, a request is basically sent out to all of the other databases that are connected through the Matchmaker Exchange. And at the base level, what we're looking for is other patients in these other databases who have the same novel gene reported as a candidate for that patient. And so what it looks like for me as a matchmaker, when I contribute a patient to the database that Care4Rare uses, which is PhenomeCentral, and we submit that patient for matchmaking, I will get a list of other submissions back from the Matchmaker Exchange from all the other individuals who have the same gene of interest mentioned.
Interesting. So I guess once you have a match, what does it mean to have a match? And how is that match going to contribute to the rare disease research?
So that's a really good question, because I would say that there is a difference between that step that I just mentioned before, which is when you get a list of cases with the same gene mentioned, and having a bona fide match. There's a bit of steps in between that have to happen. So as I mentioned, we're really looking for patients that have a similar set of phenotypes and a similar type of variant. So in order for a match to be considered of interest, we first need to do that check to see how similar are these presentations and how similar are the variants that we're seeing in each of these patients. With that said, once we have a bona fide match through matchmaking, this can sort of be the start of collaborations that can help push research forward for those patients and for that particular gene. So for example, finding similar patients allows us to build cohorts of similar patients that we can publish as a cohort. Knowing similar patients out there in the world will allow us as well to perform functional work. So we can look, say, in the tissue samples of multiple patients to see how is that gene disrupted in their tissue samples, and how does it affect certain biological functions. And then lastly, knowing multiple similar patients who have different variants in the same gene might allow us to study these variants further in model organisms. So for example, if we had a couple of variants in a gene that we're interested in, we may be able to study multiple variants like that in a mouse and see whether that mouse generates similar features to what we might see in our patient.
That is very inspiring and actually gives me a lot of ideas of how to plan the research. So what does it mean to evaluate the match due to the lack of phenotypic or inheritance information?
I think this question really speaks to that process I was just talking about, where you're trying to get a bona fide match, a true match for your patient. It really involves looking at the presentations of the two patients and the variants of the two patients. When we first get notification of a match through the Matchmaker Exchange, I'm given whatever information has been put about that patient into the matching database. So sometimes I will get variant details, sometimes I'll get details on their clinical features or phenotypes. And sometimes this information is enough to say that this match is really of interest, or that these patients really don't match each other. Maybe their presentations are too different, or the types of variants they have are completely different from one another. However, most of the time in our experience, there's generally not enough information that's initially exchanged to really get a sense for whether a match is of interest or not. And so for these cases, I often need to follow up with whoever submitted the other case and exchange more information by email to get a sense of whether these patients are really a good match for each other or not. So that's sort of what we mean by we need to evaluate that match further.
I see. Does this happen often in the process of matchmaking exchange?
Yes. Yeah, I would say it's very, very common to have to not have enough information up front to evaluate these matches effectively. And so we actually recently published a paper documenting our 2 years of experience with matchmaking, and that's sort of what we saw, was that most of these matches require follow-up by email to really discern whether they're good fits for each other or not. So I'd say it is a common thing to have to follow up by email.
That sounds very challenging to sort through a large amount of information and find to actually validate that. What are some other major challenges or difficulties you face in the process of matchmaking?
Well, so I think ultimately, although it can be time consuming to do these follow-up by email, I think we still consider matchmaking to be a really successful tool in helping us get evidence to solve undiagnosed rare diseases. So generally in our experience, about 15% of the time that we submit a case for matchmaking, we're able to find a good enough match that leads to some kind of collaboration in the future. I think because of the emails that often need to be exchanged, it is a very time-consuming process, and that becomes even more true as the number of submitted cases in the Matchmaker Exchange continue to grow. Again, there's over 120,000 cases in there currently. I think another challenge is related to the accessibility behind matchmaking at this point. So the current process that I've talked about, it basically involves me on my end having a gene of interest that I am going to submit into matchmaking. And it also requires that whoever I'm matching with, they also have to think that this gene is a candidate for their given patient. So, this can be difficult if, for labs or databases, for example, where they don't have the resource to identify those candidate genes. They may struggle to participate in current matchmaking approaches because you really need to have that candidate gene identified beforehand.
That's interesting. Is the process of identifying the gene through genetic sequencing?
Yeah. So how we do this at Care4Rare is for some patients who have had exome sequencing done, we're actually taking the data that was generated by the clinical lab, bringing it back over to the research side, and having a second look through where we have a more detailed look at genes that haven't been associated with human disease before. So that's one of the ways that we can identify these candidates, is by reanalyzing data that was produced on the clinical side. And then the other way that we can identify these novel candidate genes is through, for some of our patients, we're performing other kinds of genetic testing that isn't commonly available to these patients clinically. So that could be genome sequencing, RNA sequencing, deep sequencing, long read sequencing, etc. These other novel analyses can produce the candidate genes that we then submit into matchmaking.
Wow, thank you so much for sharing that information. So what are some of the benefits of being matched to as a potential patient, and what are the future direction of matchmaking research?
So for the first part of your question, I think it comes back to why we perform rare disease research in general, and what are the benefits to patients. I think in rare disease research, we often talk about the need to reduce the diagnostic odyssey. So the idea that when patients are undiagnosed, they may go through a long journey in the healthcare system where they're seen by many specialists, and there are many clinical tests ordered to try and get a sense for what the underlying cause for their rare condition is. And part of our research is to try and put a gene to that condition. So to understand, what the underlying cause for these features are. In rare cases, knowing this gene can have impacts on treatments. I know of one or two examples where that has been the case. But even if it doesn't change the management for that patient, we've heard from families that just having a gene known to be associated with their condition can have a major impact on its own. It's something that they can mention to family members as for why these features are happening, or something that they can tell to other support systems. To your other question about the future directions of matchmaking research, I think, as I mentioned before, some of the big challenges we have now are about the time-consuming nature it takes to exchange emails when we're uncertain about how good our matches are, and the fact that you really need to have some kind of analysis done on the research side to identify these candidates for matchmaking. One of the things that we've been looking at is developing a one-sided approach to matchmaking. And so what I mean by this is, instead of a two-sided approach where both sides need to have the same candidate gene in mind, this one-sided approach would be, on my end, for example, I have a patient with a variant in gene X that I'm really interested in, and I would submit that gene into a matchmaking network, and it would search through the raw genome data for all of the patients in the database to find variants in that gene. So it doesn't necessarily rely on having that patient have a candidate identified. It allows us to just sort of look through that genetic data agnostically to try and find potential matches.
That's wonderful. So it sounds like it would be you making the judgment whether if that gene is a match, instead of having two people making that judgment.
Yeah, and I think it won't completely eliminate the need for follow-up emails, but I think by having more detailed information provided up front about these matches, it'll allow us to filter the potential matches much more efficiently, and allow us to reach out to the ones that really could be a good fit for our patients.
That's wonderful. So I have one last question out of my personal interest, and everyone out there that's interested in genomic analysis. Mm-hmm. So what is it like to work with genomic analysis? And I guess it's two questions, actually. The second question is, what is the future potential in the field that you see?
I mean, so I've really loved working in genomic analysis, and especially how genomic analysis relates to rare diseases. And part of that is, I think, because of the impact that it has on the patients. As I mentioned, a lot of these patients who are undiagnosed go through seeing many different specialties and having many different tests done clinically. And so for some of these families to get to find an answer after years of going undiagnosed, you can really see the impact it has on their individual lives. From the technical standpoint, I find genomic analyses are just really interesting because it keeps you at sort of the forefront of genetics research. Over the last 10, 12 years or so, exome sequencing has become more and more ingrained in clinical practice. But on the research side, we're starting to explore some more cutting edge technologies like genome sequencing, RNA sequencing, and long read sequencing. So those are some of the things I really like about being involved in this field of research. In terms of where the potential for the field is going forward, I really see it as taking a lot of these novel approaches that I've been talking about and sort of combining our analyses together. So we know that genome sequencing is very good at picking up certain types of changes. So we can see large rearrangements within genes, for example, or we can see variants that are outside of the coding regions for genes. On the other hand, RNA sequencing can give us information about how much activity certain genes have, or how the splicing of certain genes are changed. And so I think the future is really going to be, how do we look at these large sets of data together in unison when we do our analysis? Because I think it'll provide a more cohesive picture to help us figure out what exactly could be the cause for an undiagnosed patient.
That's really, really exciting. Thank you so much for the incredible research that you share with us today. And First of all, this is absolutely inspiring to me, and I have known about the blooming of genomic analysis in the field in the past few years, but this is my first time learning about the amazing application of genome studies to rare diseases. This exciting result is very encouraging, and moreover, the data sharing opens door for a global rare disease community to be more connected, even helping each other to advance in improving understanding and treatment in patient care. So I'm extremely grateful for the great work you and your research team is doing, and I'm so happy to learn from you today. So thank you again, Matthew, for giving us the time of your day, and I'm excited to learn about your future research as well.
My pleasure, Son. Thank you very much for having me on again.
Thank you.